agenda. So for today, I will start with a welcome. So first of all, I want to welcome all to this kind of a new seminar series from the Basic Science Core from the DC CFAR that we've started to organize. And the objective of this seminar series is to highlight junior investigators within the DC CFAR who had made an impressive impact on the HIV field. So every year we're looking at the publications and select what we think are the most important advances of HIV research from the basic science point of view. So it's my privilege and honor to introduce our first speaker for this seminar series, Dr. Rachel Resop. Dr. Rachel Resop had a BA, a double major in both in biology and in Spanish. So I know that she is fluent in Spanish. It's something that we both have in common. So at the Ripon College, after that, she moved to UCLA, where she obtained her PhD under the direction of Dr. Uten Bogert to characterize it with a dissertation title, Characterization of Sphingosine 1-phosphate signaling in the human thymus, implications for T-cell reconstitution in HIV infection. Rachel continued her studies for a couple more years after finishing her PhD in the same laboratory with an NIH T32 fellowship. Upon that, in around 2008, she made the wise decision to join my lab. Maybe it was a wise decision or not, that's to determine, but where she's been working on the role of sphingolipid signaling in productive and latent HIV infection and in cell-to-cell transmission, where she will be presenting some of her work today. So over the years, she's been really successful in obtaining different fellowships as well as traveler scholarships to go to different meetings. And most recently, actually, she, unfortunately for us, she will be leaving sooner than later, hopefully in the later than the sooner, because she was recently awarded a postdoctoral fellowship from the Fogarty Global Health Research in a collaboration between UNC and Lusaka in Zambia. So she will actually be moving to Zambia, where she will continue some of the studies on the role of sphingolipids in HIV infection. And this is actually something that she's really passionate because it brings what is her other passion, that is global health. In fact, Rachel is being really active on pursuing further education on that regard with a certificate in public health from the George Washington University, and as well as she will continue to obtain a master's in epidemiology from the London School of Hygiene and Tropical Medicine, while she is doing her next postdoc in Zambia. So with that, I will just let Rachel to present her work, and I hope that you all enjoy it.
Thank you very much, Alberto, for the intro. I appreciate the kind words, and trust me, it will be difficult on both ends for me moving on from your lab. It's been such a positive and stimulating work environment. So I've really appreciated that the last 3.5 years or so. It'll be about 4 years in October. So I'll go ahead and share my screen. Just give me one moment, please. And I'll also stop my video if that's okay with everyone while I present, just to ensure that my connection is steady and fast. Okay. Sorry about that. And apologies in advance if there are any helicopter noises. I live in Capitol Hill now, and we're right on the flyway for Biden and for everything going on around here. So it's interesting. So I've changed the title of my talk a little bit just to reflect that I'll be presenting both the paper that was advertised for the seminar series, as well as a little bit of new data that we're currently working on to sort of continue this project and look at the next steps for the big picture of sphingosine 1-phosphate in HIV infection. And so again, I'd just like to thank the organizers for having me. It's an honor to be presenting today, and thank you for your attention. Please feel free to stop me at any point with any questions as I'm going along. So I thought I would start in light of the fact that we're now in the 40-year anniversary of dealing with human immunodeficiency virus, or HIV, in the world. I thought I'd start with this kind of throwback to the first reports that came out from the CDC June 5th of 1981. So I actually didn't know until this year that the anniversary of this report was my birthday. So when I found that out, it's not my exact year of birth. I'll leave that to the imagination of everyone. But when I found that out, I was actually kind of I thought that was kind of cool, because I thought maybe it's meant to be that I ended up as an HIV researcher. But it also hits kind of hard, because now we're in 40 years of trying to combat HIV. And this is just to kind of refresh us all. This is one of the first reports, and it was 5 young gay men who, at that time, they didn't know what it was. And this later became HIV. And so now we're 40 years into it, and we're continuing to work on trying to combat this virus, and it remains a global health issue. Here are a few stats that I just pulled from the UNAIDS site. So they have a lot of interesting information if you'd like to see more. I don't think that all of us are in the HIV field, so I just thought it would be a really a great reminder that since those 40 years, since the start of the epidemic, we've had 77.5 million, could be more, this is the confidence interval, people who have been infected with HIV. And currently there are almost 40 million people living worldwide with HIV. 1.5 million became infected last year. 690,000 people died from AIDS-related diseases in 2020, and 27.4 million are currently assessing antiretroviral therapy, ART. So compare that to the number of people living with HIV, and we can see that we're doing a good job, but we have really a long way to go. And there are a lot of disparities as far as global locations of where people are able to access ART and where they are not. Currently available ART focuses on targeting multiple stages of the HIV-1 life cycle, for example, reverse transcription, entry, and fusion, and treatment of HIV is lifelong due to establishment of latent infection. So this refers to the fact, for those of us not in the HIV field, that HIV can integrate its genetic material into the genome of the host cells. And those cells can become resting, and then at a time in the future, they can become activated. And this results in a new wave of production of HIV. So it's really a lifelong sentence. And we do a pretty good job with the ART regimens that we currently have available, at least with the people that we are reaching with ART. But it is a lifelong commitment to taking ART, and we're currently looking at whether there are some other potential therapeutics that may be worth investigation. Few of the potentials have really focused on immunomodulatory compounds that may harness the immune system to combat the virus. As I mentioned, most of them, it's great that they're specific. They target specific stages of HIV-1, but it's always good to keep an open mind. And few of these have really focused on an actual component of the immune system that we may be able to harness to fight HIV. So we wanted to explore this idea a little bit further in our work, as well as also try to understand more about how host immune factors are involved in progression of HIV infection. So I'd like to actually start with a little bit of a summary slide. So this is kind of an overview of what we'll be talking about today. Alberto created this with BioRender, and we meant to use it for a striking figure that they requested for the paper, the fingolimid paper, but they ended up not, plus pathogens ended up not needing it. So I'd like to present it here today. So this is the HIV life cycle. So I'm not sure if we're all in the HIV field, so just a quick overview. This is a schematic of an HIV virion. So HIV binds to host cells. Then it undergoes fusion with the host cell membrane. It's taken into the host cell. And there are multiple processes that happen, including reverse transcription of the viral RNA into viral DNA. So now you see double-stranded DNA. It's taken into the host cell nucleus where, as I mentioned, can integrate into the host genome and then undergoes transcription and translation, export from the nucleus, and assembly of viral particles. So here you see the core, the virus core, and everything's coming together. Then there's budding, and new variants are produced from the host cells, taking little bits of the host membrane and other host components with during release. So that's just a very, very basic overview. It's a complex process. And so we actually were only really able to focus on this half of the schematic for the studies that I'll be showing you today. And for this, we wondered whether or not targeting a host component, that component being sphingosine 1-phosphate or S1P, could affect any of these different stages of the HIV-1 life cycle. And could that really play out into a reduction in productive or latent HIV infection? So in order to do that, I'm not showing S1P here, but we are showing FTY720. This is also called fingolimod, and this was kind of our star player for the studies that I'm going to be showing you right now. So FTY720, or fingolimod, as it's known when used in the clinic, is a functional modulator of the receptors for S1P, and I'll be telling you a little bit about that coming up. So we wanted to look at in these studies, if we use this compound and we inhibit S1P, can we block any of these stages of the HIV-1 life cycle? So what is S1P? Well, sphingosine 1-phosphate, which I have some previous experience working on from my dissertation work in a very different field, in basically in T-cell development in the thymus, is ubiquitous in the body. It's a sphingolipid chemotactic signaling molecule, but it's involved in a myriad of processes, including just regular cell signaling, which varies depending on the receptor that it binds to, cellular life or death decisions, chemotaxis, and many other functions. It's been well studied in cancer as well as in other infections, but not really in HIV, with the exception of these few papers that I'm showing here on the right. Around the time that I joined Alberto's lab, late in 2017, or a little bit before that, this paper came out from the Francois group in Canada, I believe, and they looked at whether we could target S1P, components of S1P signaling to affect a function in reversal of HIV latency. So they found that ligands to S1P receptors may actually function as LRAs, or latency reversing agents. And I think this paper really caught the eye of both Alberto and me, because Alberto is an expert in HIV latency, and I was interested in S1P. So we kind of bonded, excuse me, over that paper, and that kind of inspired a lot of the early studies that we did together. Then a little more recently, in 2019, Pino et al. From the Pierdini group published that fingolimod actually has an effect on retaining cytolytic T cells, and it limits TFH cell infection in certain lymphoid sites of SIV persistence. So they looked in macaques, and they saw that if they modulated S1P signaling, they could actually limit infection in their model. And we also saw this presented at CROI. So that was really interesting. And at that time, we were already working on S1P signaling and HIV. So we kind of started to see a little bit of a picture coming together here. So again, it's a ubiquitous signaling molecule. It's involved in so many processes. So how to go about targeting this to see if we could affect HIV infection? Well, just to give you a little bit of an idea, this is, sorry, it may have got a little bit blurry. This is an overview of the S1P signaling system. So sphingosine 1-phosphate, or S1P, is depicted with these little red spheres here. And it binds to one of 5 S1P receptors. So those are called S1P 1 through 5. So at least that part is simple. But the straightforward nature of S1P signaling stops there, because depending on the receptor that S1P binds to, the signaling, downstream signaling actually varies. So these are G protein coupled receptors, as you can see depicted here. And depending on the receptor, which depends on the cell type, as far as the expression of the receptor, the downstream signaling varies. So that may involve immune trafficking, Th1 or Treg differentiation, Th1 restriction and modulation, cell development, etc. Now, some of these other receptors are more important for macrophages or DCs, for activation of those cells, for inflammation, the endothelial barrier, and even NK cells, which our lab is now working on as well. So that's S1P5. So it's really fascinating that the binding of S1P to the different receptors can have such a wide range of effects, just depending on the cell type and cell receptor expressed. And some cells express more than one S1P receptor. For example, we and others have shown that CD4 T cells express S1P1 and S1P4 to the greatest extent, and maybe a little bit of some of the other S1P receptors. So fingolimod, which I'll be talking to you more about for the rest of the talk, is a functional modulator of S1P 1, 3, 4, and 5. And some groups have reported it may also be modulating to a limited extent S1P2, depending on the cell type, etc. So it's not a very straightforward picture, but it is very interesting to study. And then just on this end, I'll just briefly mention and then talk more about it at the end of the talk. S1P is generated by phosphorylation of sphingosine to sphingosine 1 phosphate. And that task is completed by sphingosine kinases, of which there are two isoforms, sphingosine kinase 1 and sphingosine kinase 2, or SFOOK 1 and 2, depending on how you want to pronounce that. It can get kind of fun if you play around with it. So we'll talk about that more at the end. For right now, we're focusing on inhibiting the receptors. But the next step, which I'm working on now in a follow-up paper, is inhibiting the availability of S1P itself by blocking the sphingosine kinases. So I hope that made sense to everybody. And this slide, I forgot, had animations, is just to mention that sphingolipids, it's even more complex than that, because sphingolipids are also components of the cellular membrane. They maintain ceramide, maintains the integrity of the membrane in many cells, and there's also conversion of sphingosine and ceramide that occurs there. And S1P receptors, again, they're present at the cellular membrane. They're 7 transmembrane receptors, G protein coupled receptors. So there's really a lot going on with S1P signaling in almost all of our cells. If you think about it, it's pretty fascinating. Interestingly, in 2004, so a while ago, Puri et al. Found that if you block glycosphingolipids, you can inhibit HIV entry. So with that in mind, we thought, okay, there's probably something going on here, either with the S1P receptors or S1P itself, given that it has so many functions in cells. So we really wanted to investigate how that may be affecting the progression of HIV infection. So in order to look at that, we decided to utilize the primary cell model of HIV developed by Alberto many years ago during his postdoc with Dr. Planeas. And since then, this model has been used extensively to look at many different questions related to HIV latency, the effectiveness of different LRAs, and many other interesting areas. I have yet to read all the papers. But in order to look at whether fingolimod, or inhibition of S1P receptors, had an effect, we decided to use FTY720 at many different points of this model and examine different stages of HIV infection. So the model is, this may be review for some people present, is generated by isolation of naive CD4 T cells by negative isolation. That's day 0. We then activate the cells and expand them in the presence of IL-2 for a number of days. At day 3, we activate for 3 days and then we expand them, just to clarify that. At day 7, we do a spin infection, generally has been with NL43, but our lab in general has been using many different viruses, including JRCSF, NL88, and even founder viruses to infect. We then infect for 3 days. I'm just saying NL43 here because the majority of what I'll be presenting is with this molecular clone. After 3 days of infection, we crowd the cells, and we do this in 96 well plates, always in the presence of IL-2, and now they're infected. And the crowding is really to facilitate cell-to-cell transmission, which we notice that from day 7 to day 10, there's a small percentage of cells that are infected, but then from day 10 to 13, that really grows. So the closeness of the cells facilitates the transmission of the virus. We then uncrowd the cells, and we usually add ART, depending on what version of the model we're doing, but usually we carry it out until day 19. So at day 13, we add antiretrovirals, and I've usually used raltegravir and nalfinavir, but other people have used different ART regimens. And this is basically to reduce the productive infection, but it will maintain whatever portion of cells are becoming latently infected. And then at day 17, we isolate the CD4 positive fraction of cells. And we do this to isolate the cells that are not productively infected, since HIV downregulates CD4. So that proportion of cells that we isolate, that population, now contains some cells which may be latently infected, which usually are some cells latently infected, and some that are not. So from there, we reactivate in the presence of a variety of LRAs. The control for reactivation in our model is anti-CD3, CD28 stimulation with beads. We also use IL-2 as a weak LRA control. And then other people have used a variety of different LRAs, and I'll touch on this a little bit toward the end. Then at day 19, we assess P24. So that's the intracellular HIV P24 GAG protein, and we do that by intracellular flow cytometry. So I hope that was a good overview for everybody. And in order to look at FTY720 and the effect on HIV, we used it at different stages of the model. So I'm just showing here as an example. A lot of my experiments have been treating from day 10 to 13 with FTY720. And so I'll also show you data from treating earlier, so pre-treatment, before infection, and we'll look at later on, so reactivating in the presence of FTY720. So the first thing we wanted to do was determine whether or not FTY720 was safe and effective, at least in our in vitro model. And we did that by measuring viability dye, which binds to the membrane changes during the start of cell death, and then also caspase-3, an early marker of apoptosis. So we looked at viability dye and activated caspase-3 after 72 hours of treatment or no treatment with FTY720. And we saw that there was really no change in the viability dye, excuse me, or activated caspase-3 population following 72 hours. And this was uninfected cells, which you can see summarized here. We did several donors. We then looked at the viability during infection with either NL43 or NL88. So this is an X4 or R5 tropic HIV molecular clone. And we looked at a variety of concentrations of FTY720. So we wanted to determine which is the most effective and which is safe to continue using for further experiments. And we saw that really, with the exception of one donor here, which was a little more sensitive, as well as this interesting donor, which had lower viability overall, there was really no effect on the viability of infected cells with FTY720. So they were pretty much at the same level as the untreated infected cells. So we ended up using, for some experiments, the full range of concentrations, and then for some of them, we zeroed in on the kind of the higher end, since it seemed to be having more of an effect and it was not causing viability problems. So the first thing we wanted to see after determining the safety of FTY720 was whether or not a pre-incubation with FTY720 followed by infection would have any effect on the level of HIV infection, which is almost always assessed here by P24. So what we did was we did a pre-treatment for 48 hours with 30, 44, 66, or 100 nanomolar FTY720 for 2 days. So we pre-treated, again, this is the same model, I'm just showing kind of a little abbreviation of it, since this is the readout for these experiments. We pre-treated from days 5 to 7 with FTY720, and then we infected with either NL43 or NL88, so either an X4 or R5 HIV molecular clone. And for those of us not in the HIV field, that just refers to which co-receptor the virus is using to enter cells. Please stop me if there's any questions about anything. So, so following the pre-treatment, we had infected at day 7 after 2 days of pre-treatment, and then we left them for 3 days in culture before assessing the P24. So this is just an example of one of the donors. This is an example of an NL43 infected donor. So here's uninfected, HIV untreated, and then 30 all the way up to 100 nanomolar FTY720. And we can see that here's a summary of that on the right, we can see that treatment with FTY720 had a pretty nice dose-dependent effect on reducing the percent P24 CD4 negative cells. And again, that's just the non-productively infected, because the productively infected, as you can see here on the flow plot, they downregulate CD4. So this is infected. So the ones that are P24 positive have downregulated CD4. And we saw the same irrespective of the tropism of the virus that we used. So NL43 here, NL88 here, and we really saw a dose-dependent effect. Sorry about that. A dose-dependent effect for both molecular clones. So from that, we got pretty excited because we saw that, okay, treating with FTY720 does, in some capacity, reduce HIV infection in CD4 cells. So we next became curious as to whether that was an overall effect in the culture, like maybe could have to do with viral release or some other effect overall, or whether it was specific to the target cells. And by that I mean, was it specifically rendering cells less permissive to being infected? So in order to look at that, we set up a co-culture system, and we labeled cells that were either untreated or treated with FTY720 with cell trace dye. And this is kind of similar to CFSE, if you may have used that in the previously. So we had either untreated or treated, and we were able to track those cells in the co-culture. So we pre-treated for 48 hours or not, and then we added to pre-plated infected crowded cells. So to clarify that a little bit, we had cells that were already going through the whole model. They were not treated. They were infected, and they were here at day 10 to 13, which you can see denoted up here. And then at day 11, for 48 hours, we added those pre-treated cells into the co-culture. So please just ignore this treat here, because that actually refers to the pre-treated cells. So we had now a co-culture which had cells that were pre-infected, and then we had added to that treated or untreated cells, and we were able to see what happened to those treated or not treated cells. So what we saw, if you compare these are all cell trace positive, so these are those target cells. What we saw was that the untreated had, in one donor, this is just an example, had about 6% P24, and then the treated had only a third of that. So that was reduced by quite a bit. And then here's another donor which started out at a lot higher of infection. So these are the untreated target cells, and these are the treated ones. So it was, in both cases that I'm showing here, it was really only about 1/3 the level of the untreated condition. So FTY720 treated cells were reduced, had quite a bit reduced P24. And from that, we were able to understand that this was really quite a specific effect to the target cells. So we also looked as a control at the other cells, the kind of the feeder cells, if you will, in the system. And we saw that there was a little bit of a reduction overall in P24, but the P24 in the feeder cells was really quite consistent, whereas it was only reduced in those target cells. So this seemed to be really a specific effect of the target cells, and probably not as much on viral release or some other factor. So we then wanted to look at what happens to productive infection overall. So if we add during crowding, so during days 10 to 13, during the crowding of infected cells, what happens to the level of productive infection? Well, we saw, if you look here at the raw values, we saw that there was a decrease in all donors that were treated with FTY720 in productive infection. And again, this is just assessed by flow at day 13. And we saw the relative infection was also decreased. And this is just how we determine relative infection. We just normalized by looking at the difference between the day 13 treated and day 10, divided by the day 13 untreated minus day 10 and multiplied by 100. So this is just our way of looking at normalized infection. So that was decreased. Okay, so then what happens in latent infection? So then we carried the full model out to day 19. And again, we had treated during days 10 to 13 during crowding. So we wanted to see if we treat during crowding, does that affect carry out all the way to the reactivation of HIV? So indeed it did. This was a very interesting donor that had an exceptional amount of reactivated HIV. Most of them for me, are more in this range, sometimes lower, 2, 5, 6%, something like that. But all of them had reduced reactivated virus when treated with FTY720 during days 10 to 13. And this is just the normalization here, which we did in a similar way to the day 13. So from that, we've seen, okay, there's an effect when you pre-treat. So there's an effect on cell-free infection. There's an effect during cell-to-cell transmission during crowding. And that effect carries out, these were the same donors, all the way to the reactivation. So if we treat during crowding, if we treat during cell-to-cell transmission, we can, in effect, reduce, and I'm realizing I'm going to say a controversial term here, the seeding of the latent reservoir. So you may argue with me on that, and I'm happy to rephrase my, my wording. But we were basically reducing the, the pool of the virus that was going to be reactivated, the pool of infected cells. So we then wanted to start digging a little bit into the mechanisms. So we collaborated with Remy Fromentin and Holly from the lab of Nicolas Chamant. And using our samples, they performed a total and integrated HIV DNA assay. This is a PCR-based assay, which is a nested PCR detecting first the human genome, so human CD3, and then detects either the total HIV DNA with primers called ULF1 and UR1, or the integrated virus ULF1 and ALU sequences. So working with them, we were able to determine that when you looked at those cells from day 17 prior to reactivation, there was reduced total HIV DNA, and this is just the raw values, excuse me, and the normalized, and also reduced integrated HIV DNA, again, just the raw values and the normalized. So we saw that from this, there was some effect that was happening at or prior to reverse transcription. So if you'll think back to that first schematic that I showed with the different stages of the life cycle, we were able to kind of narrow it down at this point that, okay, there's something happening at or prior to RT because we have reduced integrated as well as total DNA in cells. So then we thought, okay, maybe there's an effect on the fusion or the binding or the fusion or the entry of HIV. So again, we collaborated with some colleagues here at GW, with Larissa from the lab of Dr. Bukrinsky. And with her help, we performed the NL43 BLAM assay. So this assay, the method you can see here by Kavra et al., as well as others, this assay looks at the infection of cells with NL43 BLAM, which is a construct which contains the VLAM BPR. And so when the NL43 BLAM enters cells, and BLAM is released into cells and then cleaves the CCF2 substrate, and you can detect the uncleaved and the cleaved substrate by flow cytometry, which we're showing here on the left. So here you can see, again, we treated for 48 hours with FTY720 prior to infection, similar to the previous experiments. And if you look at the untreated, this is just our gating of the uncleaved and the cleaved. You can see here's the infected population for the cleaved substrate, which is what you can see here going on. And then with FTY720, in this donor, there was really not much of an effect. There was a very moderate reduction in the cleaved CCF2, indicating that there was a similar but very moderately reduced fusion and entry of the virus with the FTY720 treatment. However, in this donor, we saw quite a bit of a different effect, and we saw that it was actually quite a bit reduced with FTY720. So these are kind of two extremes, and then the majority of the donors that we did fell somewhere in between, probably closer to the moderate effect side of things. You can see that summarized here with both the raw data as well as the relative, the normalized data. So we saw a moderate effect on fusion and entry. And then we also just looked at, before that, we actually just looked at binding. So to do this, we just incubated treated or untreated for 48 hours with FTY720 cells with NL43. And then for 30 minutes, and then we just immediately lysed and performed ELISA just to see how much virus was actually sticking to the cells, or it's actually binding. And we saw that there was a moderate effect with treatment of FTY720. So overall, a moderate effect at this stage of the life cycle. And then to complement that, we went ahead and looked at the MFI, or the mean fluorescence intensity of CD4. So this reflects the density of CD4 on CD4 cells. And we saw that with FTY720, that was also reduced. So that made sense in the context of reduced fusion of the virus because there was less CD4 present for the virus to bind. We also saw a reduction in the MFI of CCR5 on the same cells, but not much of an effect on the CXCR4 co-receptor. So there may be a bit of an effect here, but the CXCR4 doesn't really seem to be playing into it. We next looked at whether there was an effect on the cell cycle state, because it's known that depending on the state of the cell cycle, cells may be more or less permissive to infection. So to look at this, we did both a flow-based assay for cell cycle state, as well as looked at Ki67, which is a marker of proliferation in many different cell types. So we noticed that when we treated with FTY 720 from actually 72 hours, but looking at day 10, 11, 12, and 13, we noticed that there was an effect on the cell cycle. So here you can see pyronin Y staining on the Y, that's detecting RNA, as well as 7-AAD, which detects DNA. And we saw that we started out the same here, as you can see, and then by day 11, with treatment with FTY 720, there was a bit of a reduction, actually quite a bit of a reduction in the SG2 phase, which I apologize, I don't have that labeled here, but this population basically reflects the cells which are cycling. So this is SG2. And then there was started to be an accumulation of cells in the G0 phase, which is here in this little box. And that's noticed to a greater extent by day 12, so 48 hours later. That was really quite a bit different, from 7 to about 2.9% SG2. So really a decrease, and then increasing cells kind of piling up in G0. So you can see that summarized across the several donors that we did here. G0 is actually increasing quite a bit across the board. I apologize, it seems that the legend has actually somehow got lost here, but the gray is just the untreated and the green is the treated with FTY 720. So that's increasing, especially by 48 hours and out. Not much of a change in G1B, but then SG2 is decreasing quite a bit with FTY 720, especially by 48 hours. So that's kind of the time point that we were doing for a lot of the experiments, 48 to 72 hours. So that's fitting with what we've seen with the effect on HIV P24. And then we saw a similar effect with Ki67. So with 48 hours of treatment, excuse me, at 2 different concentrations of FTY, we saw quite a bit of a decrease in the percent Ki67, kind of across the board. So I could have given each of these a unique symbol, but this is generally that each donor had a decrease in Ki67. So to dig a little bit further into the mechanism, we wanted to see if there was any effect on an innate HIV-1 restriction factor known as SAMHD1, which is known to have an effect on restricting HIV by limiting the DNT pool in cells. So limiting the raw materials that the virus needs to replicate. So we looked at SAMHD1 phosphorylation. So the phosphorylated form of SAMHD1, or pSAMHD1, is the inactive form. So a reduction in the inactive form with consistent expression of the total form would indicate relative increase in SAMHD1. And that's actually what we saw with FTY 720. So here are 2 different example donors. Here's a treated and an untreated for 2 donors. And you can see that pSAMHD1 is decreased with FTY 720 in both donors to different extents. It was really quite donor dependent. So it's just summarized here. So you can see that most donors had actually all donors had a decrease, just to different extents. And most donors had, I will say, relatively consistent, because it was really a little bit a little bit different across the board with the total SAMHD1. But when we looked at the relative expression of total, that was staying about the same, but the phosphorylated was definitely decreasing in all donors. So overall, we saw that there was an effect on reducing the inactive form of SAMHD1. And I'm not going to show it today in the interest of time, but we've now developed a flow cytometry-based assay for SAMHD1 for both total and phosphorylated. And if anybody's interested, I'm happy to share what we're using, because it's definitely a time saver when it comes to doing these kinds of experiments. And we've actually verified the effect of FTY 720. It's actually even, I will say, more clear with the flow assay, as well as you're able to detect the different populations. So it's actually pretty interesting. We verified that there's an effect of FTY 720 on reducing pSAMHD1 also by flow cytometry. So that was encouraging to see. I'll just very briefly mention this because it's a very busy slide, but I'd like to thank Daniel Newman. He's no longer in the lab with us, but he contributed a good deal to this part of the project. He did a couple of these Westerns and I did the rest. We wanted to look at a lot of different cell cycling proteins that might be, we were really just trying to find out what could be involved in connection with SAMHD1 in our model and what we were seeing. So we looked at a lot of different CDKs, both total and phosphorylated. We looked at cyclins, we looked at P21 and P27, and we didn't really see much of a difference in any of them. As you can see, there's just kind of overall not much of a difference. However, when Daniel did a Western for cyclin D3, we actually saw that there was a consistent reduction in cyclin D3 expression with FTY720. So we're not yet quite finished teasing out the full mechanism of what's going on with SAMHD1, but we think that this is probably connected to cyclin D3, especially in light of what we've seen with the changes in the cell cycle that I showed you previously. So the last thing we wanted to look at was whether or not FTY720 had any effect on the action of established LRAs. So this is important if you consider, well, let's say we want to look at this as a possible therapeutic alternative sometime in the future. But what about those people, what if we want to treat with sort of a kick and kill or a shock and awe, or however you want to call it? What if we want to also treat with LRAs at some point? Well, we want to make sure that FTY720 doesn't interfere with the action of those established LRAs. So at day 17, again, during reactivation, we treated with a panel of established LRAs from SAHA to HODHBT, which Alberto discovered as an LRA, PAM to CSK4, which Amanda has done a lot of work on in our lab, and Inganol. And in conjunction, we compared to treatment with those LRAs with FTY720. And we did not see any difference across the board in action of those LRAs in reactivating virus when we had FTY720 in culture. We saw a little bit of a difference here with CD328 with one donor. And interestingly, FTY720 on its own was a very, very weak LRA. So that was kind of interesting. It pretty much on par with our weak LRA control IL-2. So we were encouraged to see that there was no effect on hindering the ability of any of these established LRAs to do their job. So what I've shown you, and bringing us back to this summary slide that I showed at the beginning, is that S1P receptor modulation reduces cell-free, productive, and latent HIV-1 infection in CD4 T cells. And it's linked with the innate restriction factor SAMHD1, which is modulated by FTY720 in a mechanism that we're still teasing out, but seems to involve cyclin D3. HIV-1 binding and fusion and CD4 density on cells are altered by FTY720. So as you can see here, FTY720 is acting at multiple stages of the HIV-1 life cycle to hinder infection in CD4 cells. So in our next steps, we became interested in what if we modulate other aspects of S1P signaling? And I actually can't see my timer. I think I have about 5 minutes left. So someone just stop me if I happen to be going over time. Just tell me to stop. So I just wanted to show a few slides of the sort of the new direction that this project is taking. And we're actually we actually have written up and are trying to submit this publication shortly. So hopefully you all might be interested and look for it in a little bit. So what I've shown you is that if we modulate the ability of S1P to bind to its receptors using FTY720, we can inhibit HIV infection at multiple stages. So then our question was, well, what if we just inhibit the availability of S1P? So what if we block the sphingosine kinases and we force the accumulation of sphingosine and reduce the availability of S1P itself? It would be a little bit more direct, perhaps, so a different way of looking at it. So we have FTY720, which blocks the receptors, and we also have compounds available which inhibit sphingosine kinases. And here are a few of those there, right here, but there are more that actually exist. And the ones that we've used are DMS, sphingosine kinase inhibitor 2. They all have about 4 or 5 different names, of course. And this long one here, ABC, also known as apaganib, when used in other applications. So to give you a little bit of information about those, we can inhibit the sphingosine kinases using this dual sphingosine kinase 1 and 2 inhibitor, or DMS. And these are just the IC50s and the concentrations that we used, if you're interested. Sphingosine kinase inhibitor 2, which is a selective sphingosine kinase 1 inhibitor And yes, I agree that that's confusing because there's 2 in the name, but it inhibits sphingosine kinase 1. I'm not sure who came up with that, but this is the reported IC50, and we've actually titrated this out, and we found that we need to use a little bit higher of a concentration in our model, which I'll show you in my last couple of slides. And then ABC is a selective inhibitor of sphingosine kinase 2. This is the reported IC50, and we've used it at a range of concentrations. So I'd just like to show about 3 preliminary data slides. Some of it's preliminary, some of it's pretty established, inhibiting the sphingosine kinases to try to block HIV infection. So this is the same model that you've seen previously. We used the same model of HIV infection, the only difference being that all of these experiments focus on cell-to-cell transmission or productive infection. So from days 10 to 13, we're treating with modulators of S1P signaling. In this case, sphingosine kinase inhibitors. And I'm not going to show any latency data for this part. So what happens when we treat with DMS? So this is a dual inhibitor of sphingosine kinases, so sphingosine kinase 1 and 2. Well, first we looked at the viability, and we saw that there was really no effect on viability, except in a couple of donors at the highest concentration at 6 micromolar. So with that in mind, we didn't really use 6 micromolar for many of the experiments going forward. And I'm just showing you the first few experiments from the paper that we're submitting here. So this is the percent P24, and you see that that is inhibited in a dose-dependent manner with DMS, similar to what we saw with FTY 720. The only difference here is that all of it's focusing on days 10 to 13. And here you can see the EC50 that we were able to calculate using these different concentrations, as well as sort of the inhibition curve. And the EC50 was calculated to be about 2.913 micromolar. So luckily, I had been using 3 micromolar for a lot of experiments, and then we calculated this, and we just went forward with that because it seems to be very effective with no effect on viability. And you can also use a little bit higher of a concentration as well. And this is actually a little bit out of date. I have more donors with 4.5 that I have to update this figure with, and that seems very effective and not to have an effect on viability. So we can actually inhibit more than 50% of infection. With a specific inhibitor to sphingosine kinase 2, we saw a similar effect to DMS. So we might not really need to inhibit both of the sphingosine kinases, rather just sphingosine kinase 1. And we see with the viability studies, no real effect except for at the highest concentration, which we didn't really use as much going forward. Although it was very effective, there was a bit of an effect on viability. So we only went up to about 10 micromolar, which was still very effective at inhibiting infection. Here's the curve pertaining to that data. And again, the EC50 was calculated at about 10.7, 10.6, 10.7, which is right in here. So finally, what if we inhibit sphingosine kinase 2? Well, unfortunately, this one did not have much of an effect on reducing infection, which is interesting because according to some RNA-seq data, sphingosine kinase 2 may be expressed at a higher level transcriptionally than sphingosine kinase 1 in cells of our model. However, it seems that sphingosine kinase 1 might just be more bioactive or may be more readily expressed at the protein level in cells in our model. So these are studies that we have to keep in mind going forward. This is just the viability data for that. We did see that at the higher concentrations, again, there was a little bit of a drop in viability, and the projected EC50 was over 50. So we didn't even use up to that concentration because we started to see a drop off in viability. So it seems that inhibiting sphingosine kinase 2 is probably not effective at reducing HIV infection in cells of our model. I'll just very briefly mention that we also looked at the effect of the different inhibitors by biological sex. So this is becoming very important as people are finally starting to realize that biological sex is a very important variable in biomedical studies. And so we looked at FTY 720, DMS, SKI2, and ABC, and we just separated by the reported sex of the donor at the blood bank. So the caveat that I'd like to mention is that we don't know if this 100% corresponds to the biological sex that the individual was born with. So we have to keep that in mind, but we're working with what we have, which is what the blood bank gives us for each donor. But we didn't see much of a difference across the board at the effect of these inhibitors by biological sex. Interestingly, there might have been a little bit more of an effect in the males, but we're still looking into that. I'm doing further analysis comparing the relative reduction for males versus females. And I'm also completing additional donors, female donors for SKI2, because we only had a few for that compound. So that will be updated shortly. And finally, we didn't see any effect of these compounds when looking at the age of the donor. There's a little bit of a trend, perhaps. Again, this additional donors are in progress. But really, according to the R-squared value, there was no correlation at all between the effectiveness and the age of the donor. So that's encouraging, because that means that if this could ever be developed into an additional novel therapeutic, it could be used with either biological sex or any age, and it should be effective. So this is my last slide, and this is actually just preliminary data, but I thought it was kind of interesting because this could possibly be taken in many different directions. So as I mentioned, S1P is present in many different cell types, and it's also very important in macrophages, for example. S1P receptor 5 is expressed by macrophages, and S1P is important for many different functions of these cells. So Paula from our lab is actually working with macrophages for her master's project. And so around the time that she started her project, we started collaborating a little bit, and I was able to use some cells that she had, and then I started generating them myself as well. So I started to look into whether we could inhibit HIV infection in macrophages by using any of these compounds. And overall, treatment with FTY or DMS at a variety of concentrations has a pretty mild effect on reducing infection. You can see here with 6 micromolar DMS, it does reduce infection in macrophages. And these are treated with NL88, so an R5 tropic molecular clone. And yeah, there's a bit of an effect on DMS in some donors, but it's also preliminary, and this would probably have to be repeated in additional donors to really draw strong conclusions. But what was interesting was that when we generated a co-culture system using the macrophages and autologous CD4 T cells, we actually saw an effect on the transfer of virus from infected macrophages to those autologous CD4 cells. So what we did was we infected macrophages, and then we pre-treated autologous CD4 cells generated from the same PBMC donors with FTY720. And what I saw was that those pre-treated cells were less permissive to infection from the macrophages once I added them to the co-culture system. And that's pretty much in keeping with what I presented earlier, where I showed that the CD4 cells pre-treated with FTY720 were less permissive to infection when I had labeled them and looked at the effect of the target cells. So I think that's really exciting, because that means that this could potentially be relevant for the earliest stages of HIV infection, which involve infection of macrophages, transfer to CD4 cells. And it's just pretty exciting thinking of it in a little bit wider of a context. So to summarize, sphingosine kinase modulation, as well as sphingosine 1-phosphate receptor modulation, reduces productive HIV infection in CD4 cells, and there may be effects in other cell types such as macrophages. So I added sphingosine kinase here next to FTY720, and we're currently looking into the mechanism of inhibition of sphingosine kinase, because we're not sure yet if it's exactly the same as FTY720 or not. It's a pretty mysterious process with a lot of different cell cycle proteins and innate restriction factor activity as well. I'm not presenting it in the sake of time, but we have optimized the flow cytometry assay for phosphorylated and total SAM-HD1, as I mentioned, and we do see, encouragingly, and it's pretty clear that sphingosine kinase inhibition actually reduces phosphorylation of SAM-HD1. So that should be coming out in the paper. So I hope that if you're interested, you'll check that out sometime in the future. And sphingosine kinase is, as I mentioned, appears to be linked, as well as FTY720, with SAM-HD1 and cell cycle state. I'm not showing that data either, but we've shown a similar effect on cell cycle state as with FTY720. So I'm not sure if there are any community members here, but if there are, I'd like to thank you for suffering through a basic science talk. And I'd just like to sum this up in a little bit more of a lay language way for those who may not be HIV researchers. So we investigated whether we could target a component of immune signaling to block HIV infection in human T cells. And we found that inhibiting receptors to S1P, or the availability of S1P itself, does reduce HIV infection at multiple stages and by various mechanisms, which we're still investigating. S1P receptors, therefore, may represent a new type of potential therapeutic, which we and others are currently investigating. So I'd like to thank you all for your time, and I'd like to thank everybody who was involved in different ways in this project, specifically everyone from the Bosque Lab. So thank you to Alberto, my mentor, and Amanda, Pingtao, Natalie, Carissa, Callie, and Paula. I hope I didn't misspell anyone's name. This is a photo that we took during the pandemic, the earlier days of the pandemic, when we were, some of us working from home and some people in the lab with masks. And this is Ping Tao. His screen wasn't working at the moment. I'd also like to thank the previous lab members, especially Daniel, who worked with me. I mentored him a bit, and he worked with me on the SAMHD1 and Cyclin D3 projects. I'd like to thank Larissa and Dr. Bukrinsky for their help with the fusion assay, as well as Nicolas, Remy, and Holly from the Chamont lab for the integration assay, and the MITM department, all the faculty and my colleagues there, the DC CFAR for inviting me and supporting me, and the NIH funding from Alberto that supported this project. So thank you very much, and I'm happy to take any questions.
Great job, Rachel. Anybody has any questions? We have a few minutes left.
Yes, I have a question. Thank you, Rachel. Very interesting talk. Congratulations on your next move. I mean, that's really exciting.
Thank you.
I was wondering, you showed that this FY inhibitor, it modulates somehow those 7 transmembrane receptors, right? For this PA3. And I just wonder, did you look into connections between, mechanistic connections between the receptor inhibition and those biological phenomena in HIV replication that you described? How does, because I mean, maybe at least I don't know anything about these receptors' participation in reverse transcription, or I mean signaling from these receptors in reverse transcription, some HD kind of, and all the other effects that you described. So did you look into that?
As far as, just to clarify the question, sorry, as far as how the different receptors and S1P binding signaling might influence the different HIV processes?
Yes.
We haven't been able to look much further than what I've already presented. And there's not too much, sorry, there's not too much literature available as far as the basic science and the mechanisms of S1P signaling and HIV life cycle. We were kind of starting from the ground up on this. The only data that had really been available before was the one paper showing that potential of signaling could act as an LRA. And so they looked a little bit into the possible mechanisms of that. They looked at maybe TGF beta signaling and things like that, but they didn't really, that paper was not so mechanistic either. And a lot of it was done in cell lines, unfortunately. So when we try to translate that to our primary cell model, it's really hard to say whether that can be translated at all. So apart from the observation that if we modulate the binding of S1P to its receptors or the overall availability of S1P, we see changes in phosphorylation state of SAMHD1 and changes in cyclin D3 and changes in cell cycle. We haven't yet really worked out the exact signaling pathway that is causing that. Then that still has to be done. I'm not sure if that fully answers your question.
Well, yeah, I mean, at least that's what you know.
Right, that's what we know so far, yeah.
Rachel, I was wondering if the inhibitor, S1P receptor inhibitor, has any effect on the elasticity of membranes. So if you look at the bigger picture, so viral entry is affected, cell-to-cell transmission is affected, and so it's possible that the membranes are rigid, and that's how the virus mobility is affected. Is it possible?
That's a great point. I think it's definitely possible. Others have reported that the ceramides and the glycosphingolipids in the membrane, they do have functions in maintaining the integrity, which I think might be what you're getting at with, could it be more or less elastic? Could that involve the virus entry? I haven't done any studies into that myself. And I'm not really an expert in the membrane, but I think it's definitely possible. And I think it's worthy of further investigation. I haven't seen any studies on that exact question as far as entry of HIV, apart from that one that I referenced by Puri, but that was back in early 2000s, showing that if you inhibit the glycosphingolipids in the membrane, you can inhibit the the infection, HIV infection.
Mhm.
But I don't recall off the top of my head if they really looked specifically at integrity of the membrane, elasticity. I don't think they did, but I'll look back and I'll let you know what I find.
Thank you.
There may be more by now. I haven't looked into that exact, in the last year, I haven't really dug into the literature about the membrane integrity.
Hi, Rachel. Great talk. I have a quick question. Maybe you mentioned this and I missed it. So when you use your cells, because this receptor is very important on trafficking, as you previously studied this, so how HIV infection modulates the response to sphingolipids? Do you have any idea, are those cells still will be trapped somewhere, or they, have you think about some of this?
Yeah, that's a great question. Thank you. So actually in my previous life, before coming to Alberto, I actually worked on this a little bit. During my PhD, the majority of my work was on just constitutive expression of S1P receptors in the thymus and the effect on T cell development. But then at the very end, when I did that 2-year fellowship that Alberto mentioned, I started looking at the effect of HIV infection on S1P receptors, as well as the effects that might have on T cell reconstitution. So I'm speaking from a limited perspective because our lab was focused on the thymus and T cell development. So my knowledge of the signaling there is really, it's within that context. And we've been trying to have this paper published for about 5 years now. So maybe one day it will happen. But what we did see was that when there was HIV infection, which reached the thymus, this caused an increase in the S1P receptors, specifically S1PR1 in thymocytes. And it also affected the development of T cells in the thymus. So it really seemed to be forcing them to egress the thymus before they were fully developed and before they were fully trained to be out in the periphery. And this was reflective of, I believe, the increase in S1P receptor 1. And we saw this in vivo in NSG mice, which were infected with HIV. So humanized mice, sorry. So in that context, HIV seems to modulate the expression of S1P receptors. And that seems to have to do with a completely unclear mechanism, to be honest, because it actually increases the expression of CD69, which is supposed to restrict the expression of S1P receptors. And we saw that probably due to interferons, there was an overriding of that mechanism. And irrespective of the expression of CD69, as high as it got, S1P receptors were still coming up. And we saw in our in vivo studies that this resulted in a kind of an expedited export. So increased migration of T cells before they were really ready to be out in the periphery. So I'm only speaking from that perspective. I'm not sure about HIV infection in the periphery and T cell and peripheral T cells, because I wasn't working on that at all. And to my knowledge, I don't know of a paper really looking at that, but it could be slipping my mind. I can look into it a little bit more.
Okay.
Does that at all get into your question, Martha?
Yes, I was curious, thinking about if we use this as a potential therapy, would be easy to clear the reservoir, if you modulate this response to the sphingolipids. Are those cells more targetable now by the CTLs or something?
Mm-hmm.
So that's, sorry, I'm interrupting, but that is so that it was done in the SIV model. That is the,
Yep.
Where they were actually looking into that, if whether it inhibiting the egress of CTLs from the lymph node by FTY will clear the reservoir, but I don't think that they see a decrease in the latent reservoir because FTY is not reactivating the virus, right? You can maintain the CD8s there, but you need to promote the virus to be seen. So I think that was a little bit what the results on the SIV model.
So you really need to combine the blockade of the receptor with the, okay.
And for what we see, doesn't impact latency reactivation. So it could be done in the context of the shock and kill to maintain immune cells in the lymph node, for example.
Thank you.
Any other question, since we are a little bit over time?
Oh, sorry about that, by the way.
Oh, no, you're fine.
I couldn't
You're
see my
fine.
clock while I was presenting.
So if there is no any more further questions, I will say let's do a round of applause to Rachel for an excellent seminar and for starting this new series. I think our next will be in October, and we'll be providing the information through the CFAR and the newsletter. So then we'll provide more information, and we hope to continue doing this to bring this is CFAR Basic Science investigators and others that are interested in basic science, including community, into these seminars and promote our junior investigators and their work. So if nobody else has anything else, Sanjay, I don't know if or Marta, you guys want to say something from the Basic Science Core?
No, I'm fine.
Okay,
Great.
so then we'll adjourn. Thank you all.
Thank you.
Thank you very much.
Thanks,
Bye-bye.
everybody.
Where are the